of the conference on AI augmented biology. In the past 2 days, we've heard quite a bit about how AI has informed, you know, at the level of whole organisms in terms of sort of, yesterday we heard more about spatial transcriptomics at the level of tissues and organs. And today, most of the talks are going to get into a bit more detail about molecules that make up these cells. So we will have Dr. Eidecker first, who is doing some precision medicine-related AI. And then we will have 5 talks that are looking at protein structure and function and dynamics and design and all of their interactions and how AI has enabled us to get more insight into this. So for the first speaker, as I mentioned, Dr. Trey Eidecker, who is from UCSD. He has a strong interest in systems biology and has done a lot of work on defining different levels of cellular networks. Today he's going to describe how assembling digital tumors can inform precision oncology. So with that, we will have Dr. Eidecker joining us virtually.
Great, I should be good to go. Can you confirm everyone can hear me and see my screen?
Yes, we can.
Okay, awesome. Just let me know if the connection breaks up or anything, but it should be good to go. Let me get started right away and talk about what we've been doing with the intersection of AI and recognizing patterns in patient data to match those patients to drugs. Before I do get started, let me thank the organizers. It's really great to participate, even if it is virtual this time. Okay. I am trying to advance my slides. 1 second here. We're gonna try to reshare. It's not advancing. 1 second. Okay, there we go. Everything still look okay?
Yes, we can see it.
Okay, great. Yeah, something was frozen at my end. So here's the overall vision of where we're trying to get, and I put 2030 up at the top left of my screen to make the point this may not be that far out in the future. In fact, aficionados of the iPhone will note that this is not a new iPhone. It's an older image, but I think everybody will appreciate that the text and the kind of questions we'd like to ask in precision medicine of your phone or of your information system at large definitely are not yet possible in any kind of reliable way. And so the questions we have in mind would be in a cancer center setting. Dear AI, my patient's tumor, or my tumor even, has the following data frame of mutations and clinical data. What treatment should I take to best respond to therapy? And of course, there's many questions that would revolve around that key question. Now, if I'm looking at this in a drug company, maybe the class of questions would be more like the second question. Should I be looking at any new targets? And regardless of whether, which of those questions you might ask, you almost always find yourself following up with with, and why do you think that? That is to say, these systems cannot merely be black boxes that make predictions about patient outcomes, but can't defend those predictions with further conversation and discussion. And so much like you have molecular tumor boards in cancer centers that discuss patient files and decide collectively after some discussion what drug to prescribe or what treatment to prescribe, the machine learning system itself should be able to have those kinds of conversations. And those kind of conversations often gravitate towards the molecular level. And so if you say you think this patient might respond to an AKT inhibitor, for instance, then why do you think that would be an answer such as, well, this patient doesn't have a mutation in AKT directly, but they do have mutations in components just upstream in that kinase signaling cascade, and therefore they are likely to respond. That's the kind of discussions that one has. So dissecting that goal at a slightly, just ever so slightly more granular detail, from a input-output modeling point of view, rather than go direct from the patient information shown over here at left, in fact, let me get my laser pointer. So rather than go directly from the patient data, including the mutational pattern in the genome and the candidate therapies that are being considered, normally you might directly try to connect that to patient drug outcomes, such as drug responses. We're going to go through a middle layer here of molecular understanding. And so we'd like the system to first translate the input genetics and other data into impacts on molecular, cellular, and tissue organization, and then take those molecular, cellular, and tissue states and translate those to drug treatment outcomes. So that really organizes, in fact, my talk in the next 20 minutes. We have really do research in two parts of my laboratory. The first is concerned really with understanding tumor cell structure. What are the key types of multimodality data and machine learning methods we can bring to bear on those data to quote unquote fold a cancer cell and define what that structure even means. And then in the second part, I'll talk about how, as we begin to flesh out the sort of mesoscale structure of a tumor cell, how we can begin to use that in information systems to predict drug response and other outcomes. So here's, just to get right into the first part, here's where we are, and I make no claims or promises that this is the best method or the final method one would like to use. It's simply where our collective team is right now. And what I'm going to show you, to first give credit where it's due, is collaborative work between my group and Emma Lundberg at Stanford and Steve Gigi at Harvard. And it's work that was started by Yue Chen in my lab about more than 5 years ago, when Yue realized that protein measurements in immunofluorescent images, like Emma and others create through the Human Protein Atlas, is really just a very complementary but compatible type of information with information that biochemists generate at the level of protein networks and complexes, like Steve Gigi does at Harvard with affinity purification mass spectrometry. So both kinds of data use antibodies to interrogate proteins. In the one case, you light up that protein and look at its distribution in the tumor cell population. In the other case, you grind up the cell population, pour it over a column, and see what other proteins stick to your targeted protein to define that network. And what US started was finished by Clara Hu and Leah Schaefer in my lab, who scaled up the approach to look at U2OS osteosarcoma cells and the majority of proteins expressed in that cell type, about 5,200 proteins. is just over half of the detectable proteins in a given cell type. And I'll show you all the results. I'll show you in the next few slides are taken from that more recent paper reviewed to OS. So what we do is we use self-supervised learning, what would now be called, or what has been called foundation modeling. This is a case where the foundation model is essentially protein spatial coordinates measured in those two ways for each of the 5,200 or so proteins that have been interrogated. To visualize that, now of course 128 dimensions is the dimensionality of that embedding projected down from millions upon millions of dimensions in the input. That's still a lot of dimensions, and so we further projected into two to show it on a single two-dimensional slide. But in this U map of the embedding, a point is a single protein. Most times people show these things in biology. Points are single cells. Here the points are proteins. Two proteins that are neighboring in this space have similar images and similar protein interaction patterns. If you look at the highest level of organization, you're really reconstructing organelles, as shown here by simply coloring proteins by their known organellar affiliations. But you can factor any one of these regions into communities of proteins using clustering or algorithms, and do that for any one region, like is shown down here in the lower right. And of course, you can continue hierarchically that community detection process all the way down to what ends up being about 20 nanometers at the lower level resolution of this integrated data and analysis. And so here is what the hierarchy of assemblies or groups of proteins that can be detected by this integrated system looks like. We can find 275 assemblies in total from the entire cell, factoring it again into organelles, factoring those into subcellular compartments, all the way down to, at the bottom of the screen here, small protein complexes. So each one of these nodes represents a group of proteins of some size. The orange and yellow colored nodes represent protein assemblies that are pretty well known. They correspond to they align against known terms in the gene ontology or complexes annotated in databases like Corin. In contrast, the purple annotated nodes do not recognizably align to any prior cell biology knowledge. And if it seems somewhat surprising that this is just over half of what's in this library or cell map, let me give you an illustration that sort of explains how that could be. So nuclear speckles over here inside of the nucleus, inside of the cell, okay? Prior to our study, there was essentially one checkbox on human protein atlases formed that when a microscopist was looking through at these images, if she or he saw salt and pepper fluorescence inside of the nucleus, they would check the box that says nuclear speckle. This system can see 14 kinds. Many, some of which are not well described in the previous literature. Now, what do you use this repository for? Certainly, it's a nice library of cell biological objects. We'd like to, in as many cases as we can, develop these into three-dimensional structures. And with Andre Schali's group at UC San Francisco, we've been making a lot of progress there. Here's an example of one such structure pulled out of or formulated out of this map. This corresponds to the RAG regulator complex, which signals to mTOR. It's very important during cancer development. We have essentially been able to add, we've taken an existing structure of the RAG regulator and added two new components that were implicated by the imaging and the affinity purification data, BORC S6 and ITPA, using a combination of Andre's IMP software and AlphaFold multimer. What do you do, though, for protein complexes, the purple ones in that map, that did not align against any prior biological knowledge? So you clearly there don't have a structure to start from, or really any knowledge whatsoever. And so here, the co-first author of that first study, Clara Hu, had to stop what she was doing in the middle of the study and write a second paper on how well large language models were at hypothesizing new functions and structures for cellular objects. It's very similar to what you do with gene ontology enrichment, except the problem with in this case, Go enrichment is it already failed. It already told us there was no match to an existing hand-curated ontology of cell structure and function. So we're on our own. But it turns out that, as Clara very adeptly shows in this paper, this is essentially a meta-study of how well this process works with large language models, where she has some interesting controls and computes statistics on how much hallucination occurs. But we find that in much of the time, you get very good hypotheses out of large language models, whether it's GPT-4, Gemini, or Mistral, or now Claude, that as to what these gene sets might be doing. And so for this example gene set, we fed it. By the way, prompt engineering is everything here. She writes a very long engineered prompt where she says, imagine you're an assistant to exciting science lab, and you've made these exciting discoveries of proteins that work together. Here's 5 proteins that we have great proteomic evidence that they're collaborating. What do you think they do? Please give us a short phrase, like a Go term, that if you had to name a new Go term for what this process is. Please write an essay of approximately 1,000 words defending that name, complete with citations. And third, please assess your self-confidence in the first 2 answers. Please check out her paper. In interest of time, I'll say no more about it here, but it's actually a really, I think, going to be a really general study for how I think the field really now can move from sort of Go enrichment against bespoke ontologies, hand-curated ontologies, to simply talking to your chatbots. Of course, we're also thinking about ways of visualizing these maps. There, we've done a lot of work in our Cytoscape platform to visualize hierarchies of cellular objects and the underlying network and imaging data. All of these maps I've been talking about are freely available online. Now, let me transition to the second part of my talk, which is how do we begin to use these maps in cancer predictions and predictive medicine applications. I'll start by easing into this with a couple of anecdotes, and then I'll generalize. So anecdotally, as we were constructing these maps of protein complexes in cancer cells, we began to note that you saw, if you now project a set of tumor genomes on these maps, you start to see mutational convergence, not at the gene level, but at the level of these multiple proteins working together in complexes. Here's an example. This is not a new complex that we were discussing with ChatGPT. This is a well-known complex that's been recapitulated in these maps, that of collagen. As you may know, collagen is the primary constituent of the extracellular matrix, or ECM. This is against which the tumor cell population must push as it grows, and through which it must migrate if it ultimately metastasizes. This is what the Cell Biology 101 picture looks like of collagen complexes. You draw collagens in threes from a large collagen family. They braid together like hair or rope, and then that braids again and braids again to create these collagenous fibers that make up the ECM. Collagen had not previously been noted to be under any kind of mutational pressure in cancer. However, when we looked at the level of all collagens together in this complex, as shown in the lower left, and this is work done by Fan Zhang when he was a postdoc in the lab, we could see a very, very striking buildup of mutations, 50% in lung adenocarcinoma. You can, you might say, well, that's a big umbrella for mutations. Maybe that happens by chance. I assure you it does not, and Fan's done all of those underlying statistics to prove it. Now, why had it not been noted before in the Cancer Genome Atlas projects? Because those projects have gone gene by gene and looked for mutational pressure or selection at the gene level. That's what's shown here in this mutation frequency diagram. Of those collagens in lung adenocarcinoma, this is the Cancer Genome Atlas by the American NCI. 21% of tumors have a mutation in COL11A1, which seems like a pretty high number, but it turns out that lung adenocarcinoma has a very, very genome-wide, high genome-wide mutation burden, pushing even 21% into the noise. So none of these genes have been noted as significantly mutated in cancer in this disease. But when you put it all together and you look as a whole, it's very, very, very striking. Now with Stephanie Fraley's lab, we went in and laid down matrix, collagen matrices with mutations in these collagens that were observed in the wild, compared those mutated matrices to normal collagen matrices. We decellularize the collagen deposition cell line and remove it, now seed in with a common A549 lung cancer cell line and watch its behavior. As we're showing here with this Ki67 marker, the cells proliferate much more rapidly in the mutated matrix. Here's a more recent example. This is the work of Mayan Barron, a former postdoc in my lab, in collaboration with Rich White and Mohita Tagore. Rich is now at Oxford. This is another complex that we've drawn out of those maps, the desmosome, which turns out to be in striking mutational pressure in melanomas specifically. More than 70% of melanomas have a coding mutation in one at least of these desmosomal subunits. What is the desmosome? It's the cell-cell junction complex that glues together or forms the glue between neighboring keratinocytes in the skin. Keratinocytes are not the tumor cell type, that's the melanocyte. So these, it turns out, as you'll read about in this paper, it turns out these mutations are occurring likely in the stroma, not in the tumor cell population itself, which is a fascinating concept if you think about it. So we're looking really at the tumor ecosystem as a whole. But it's very, very clear in its mutational signature. Over here at the right, we're looking in the melanoma cancer genome atlas cohort and showing in red, if you look at just the ranking of genes in terms of its mutation frequency, with the highest at left, you can see all of the desmosomal genes and their mutation frequencies in red. All of them are in the top 25th percentile, all 15 are or so of genes ranked by mutation frequency. You can see in blue the well-known cancer genes in melanoma to date. So they're sprinkled right in there in that same range of mutation frequencies. So let me now move formally to the second part of my talk, and really it's more like the second fourth of my talk. I'm almost done because all I now need to I now need to take those two anecdotes and ask, how do we take those anecdotes and generalize them into a more general system for taking a pattern of mutations, analyzing its patterns on these maps of cell structure that I've been describing, moving beyond just the desmosome or just collagen complexes, looking at all of the mutated complexes in a given tumor and using those patterns as biomarkers to predict drug resistance and sensitivity to leading standards of care in cancer. Over the past 3 to 5 years, we've been pushing really hard on this concept. And I'll just point you to some of the papers that have come out in the last 12 to 18 months over here at left. Essentially, what I'm going to describe is kind of a common template for how we build these maps, or these maps and models, and then apply them to a variety of cancer therapies, starting up here with Xiaoyu Zhao's work here in cancer discovery, building models to predict the response to chemotherapies that cause replicative stress, which is by the lion's share of chemotherapies. I'll give an example in a minute from Sung-Jun Park's work on prediction of the response to CDK4/6 inhibitors like palbociclib. These are standards of care for ER-positive, HER2-negative late-stage breast cancer. Everyone gets a CDK4/6 inhibitor, but a significant number of patients either have intrinsic or acquired resistance to those. And you'd like to be able to predict that based on the input pattern of cancer mutations, and so on and so forth. The way all of these models work, though, is according to a common template shown over here on the right. So we take the patient's genome, which we list separately as somatic mutations, copy number, amplifications, and deletions. We translate those through a deep learning system shown here in the middle to predicted drug responses. To do that training, we have a lot of data now as a community in cancer cell lines. There we use resources like the DepMap or the Sanger Center, the Genomics of Drug Sensitivity in Cancer database. We can, if all goes well in the 2D cell culture environment where we have a lot of training data, we can validate and then fine tune in patient-derived xenograft or PDX models in mice, and or ultimately where we have the fewest data, but of course the clinical data that really matter in patients. Now, all of this deep learning from drug response samples and cell lines, PDXs, or patients happens in a way that's not through a black box deep learning system, but is through a neural network model where the banks of neurons or the layers of neurons in this model are directly coupled to the cellular objects, the protein complexes and compartments that were detected and identified and mapped in these structural maps I spent the first half my talk on. And so here I'm showing one of those structural hierarchies rotated upside down so that the cell itself is here at the bottom, and you blossom upwards now on this page into organelles, their subcompartments, and at the very top, the smallest protein complexes. And so you can think about the mutations in a patient as trickling down through all that structure to change the functional states as represented by the neurons assigned to each one of these complexes. So let me just give an example for how all of this works. This is the example of from Sung-Jun Park's model predicting CDK4/6 inhibitor resistance to agents like palbociclib. Here is what that structural map looks like of protein complexes that are now serving as biomarkers of drug resistance or sensitivity. The 8 red complexes listed on this screen are those that to which the model gives attention as it makes these drug response predictions. Each of them is encoded by a number of genes. And so if you want to throw away the machine learning, what the system has basically learned is a combinatorial biomarker which looks at 90 genes and their mutational patterns. But importantly, those genes are the subunits, the building blocks of these 8 molecular complexes. And so you begin to get some pathway level insight into what's going on. And here's an example in my last data slide of that. So here's one of those 8 complexes in that map. ChatGPT has suggested a name, histone-mediated transcriptional regulation. We also give a systematic name to these systems. Here is what the protein-protein interaction patterns at least look like that cause these proteins to be placed together in assembly in the structural map. MYC already was a kind of positive control of drug resistance. MYC amplification was already known to cause palbociclib resistance. But other factors in here were less well known. So lysine acetyltransferase, or KAT6A, as well as TBL1XR1 were amplified in about 8% of breast tumors and were used with a lot of attention by the model as they were strongly associated with drug resistance. Given that interpretation, given we can sort of ask the model to explain what's going on and gives it this complex, we can now go into lab and try to validate those predictions. And so what we now are showing over here in the left in 2D cell cultures, we're going to try to recapitulate drug resistance by amplifying by a mock amplification of KAT6A or TBL1XR1. So over here at the left, here is control cell population without any genetic perturbation. The upper panel are cells under no treatment. The bottom panel are cells under the CDK4/6 inhibitor. Green marks cycling EDU positive cells. They're executing cell cycle. You can see when I apply the drug works as promised and it dampens cell cycle. This is a cytostatic drug. Now look at what happens, however, when I use CRISPR-A to amplify either of these 2 factors, TBL1XR1 or KAT6A. I'm leaking through even under the presence of the drug cycling cells, consistent with the use of these by the model to predict drug resistance. So with that, I am out of time, so I'm going to just summarize in a pictorial way. This is, there's a lot of talk these days about virtual cells. This is absolutely a virtual tumor cell. It is built structurally through the integration of imaging and interaction data, or immunofluorescence and affinity purification data. Given those structural maps, we use those to guide the construction of deep learning systems, which input your tumor genome and output drug responses. And because we get coupled structural explanations for those drug responses, that is then a route to go back into the lab and perform further genetic manipulations and work I haven't talked about, but as I hope I've at least alluded to, the ability to identify new drug targets. Thank you very much.
Thank you. Do we have some questions for Dr. Eidecker? I would like to ask one. So to me, this is sort of a new concept, sort of looking at complexes as being sort of targets of drugs. Does this mean the way we should go about drug development is slightly different because now we are not concerned with a particular single mutated protein, but rather looking at the whole complex? So could there be, targeting of some wild type protein within that complex that would be maybe easier to target as a way to treat?
I think you've. I'll take that more as a comment, and I would wholeheartedly agree with your comment. Just to go back a slide. So here we found. Let's just say how that would go. So here we found a complex where we already knew MYC was involved in drug resistance in previous papers. We've now implicated through our experiments 2 additional factors. So there's different ways this could go. One really low-hanging fruit is might you be able to resensitize tumor cells to palbociclib if you simultaneously apply an inhibitor of KAT6A. It turns out that combo therapy is already ongoing in clinical trials while our paper was under review. We read about that one. Okay, so that's interesting. So there, are we using anything about the complex? I mean, we use the complex to implicate this protein as maybe a combination therapy. Maybe what you're alluding to is what I'd really like to have is I'd like to take this hairball now and really make a 3D structure or a series of 3D structural models out of it for really understanding where I can come in with small molecules or other drug strategies to perturb the structure and its function.
Right.
So really, it's like maybe if I just kind of rephrase your comment, do I want to do SAR, structural activity relationships, at the level of this whole thing, not just KAT6A or CDK4/6?
Right. Great.
Right.
We have one question here.
Hi, it's a really amazing talk. And sorry, I might miss some points. So I want to ask, can you provide some details of the deep interpretable model? So is this something like CNN or just some new architecture?
Very good question. So everything I showed is very ancient deep learning technology at this point in time. This is all, like COVID era deep learning. And it's exactly it's exactly akin to convolutional neural networks, except because they, as you alluded to, they also take structure as input, the structure of the image in terms of for every pixel as it gets mapped into your deep learning system, what your north, south, east, west pixels are, and so you're giving fields of view that are structurally related by their XY coordinates and pixels in the image. This is exactly the same as a CNN, except that it's like a graphic, right? It's also graphic neural network would be the other concept that's quite related here. The connections that you're feeding in are based on molecular connections as established by protein interactions and/or imaging. Now, what would be the modern, incarnation of this? we're certainly very interested in the use of transformer and attention-based models to replace what I've shown here, and we're working hard on that. And I'd be happy to talk about collaborating around those ideas or any other ideas people in the audience might have.
Okay, thank you.
We have one more question.
Very impressive talk, and the virtual tumor cell is a very interesting concept. And so I'm wondering, have you ever considered their interacting partners, like whether they can interact with the T cells, like if there's a mutation on some receptors like the EGFR or HER2, HER3, and whether it can affect their interactive intensity with the macrophage or T cells, and maybe we could think more ambitious, like we may have a virtual tumor microenvironment, and we could model how the patient can respond to different therapies, and like the different PD-1 or some bispecific antibodies. Do you have any ideas or data for supporting like the virtual tumor microenvironment?
So for mostly no, although I fully agree with it. I'll take your question also as a comment, because I fully agree with it, and I agree that's an important next direction. If you're interested in working on that, let me actually go back to this example. So here, please check out this Nature Genetics paper by Barron et al., because here we did, this is mostly Rich White's work, here we did have to, as I mentioned, the mutations, it looks like are actually in the stroma, not in the melanocyte tumor population. And so we had to develop co-cultures of keratinocytes with melanocytes. And so that would be maybe one source of data to begin these investigations. We have not done what you said. I think largely it's data limited right now, but I think that's absolutely the way things should go. Another person I chatted with about some of these ideas, and I should give credit where credit's due, Jinsu Ma came up, who's now at Tsinghua University in computer science there, came up with this original framework that was very much inspired by that last question. He was very inspired by convolutional neural networks. And his original work, which is a 2018 paper out of my lab, he's a former postdoc, where we first did everything I showed you in yeast, where we have an enormous amount of data compared to even cancer. So do check. He called that the D cell model, and everything I've talked about here with the virtual tumor cell was essentially based on his earlier postdoctoral work, and so he would be another person besides us to really chat about next steps. But again, really happy to collaborate. Thanks for your interest.
Okay, well, in the interest of time, I think we will call an end to this. Thank you so much for joining us from California. And one more round of applause, please, for Dr. Eidecker. Thank you. So our next speaker is Dr. Arne Ellefsen, who is coming to us from Stockholm University. So he has a long-standing interest in bioinformatics and computational science. And today he'll describe for us some work he's done on trying to fill in a gap. So what is that gap? That we know only about 5% of protein interaction complexes, and there's a lot to be discovered. So let's hear what Dr. Ellefsen has to say about that.
Okay, that works. Okay, so I don't want to hide behind these beautiful flowers. I think so. I know the cameraman has to follow me, so I'll try to stand still. But okay, I will tell you a bit about what we and what a lot of the field has done the last decade or so. And I will start actually on a time before most of you were born. Does it work? Not. For some reason, no. Okay, I'll do like this. So first time I was in China was 1991, and I took a bike from the Central Palace out to Summer Palace in Beijing, and there was no cars, it was only bikes. I guess this is how it looks today. So it's amazing to see how China has transformed during your lifetimes. And it's, I have to say, I enjoy it a lot now. I enjoyed it a lot then also, but it was very different. For some reason this one is okay. So back to proteins. So the whole idea of the great success of AlphaFold And so maybe better. It's based on the idea of coevolution. So why is coevolution important? Because most proteins are well packed. The central protein doesn't contain much water. It's well packed amino acids next to each other. But evolution is driving everything in biology. So sometimes we have a mutation. Most of them are unfavorable. If they're too bad, the organisms die, so we don't see it. But in quite a lot of cases, it's not that bad. It just makes it a bit less stable, a bit less functional. But organisms might survive, and for various reasons, they might get stuck in population. But that will have a pressure then to have compensating mutations. Of course, the mutations are random, but compensating mutations will have an advantage and survive. So they will fix these packing issues. In this case, you mutate a quadrat to a pentamer, and you have a nice packing to the new arrow again. However, this is realized in the long time ago, but people start using this for prediction in the mid-90s. But you don't also change one thing. You also change the other side of the amino acid, because interactions are not only pairwise, they're also part of a big network. And so to do this well, you need to take into account these indirect couplings also. So like A interact with B and B with C. Therefore, you see an interaction between A and C. And this is actually the first time this was analyzed and thought about. It was around the millennium, 99, by Lapidus and all. There was also another paper out there. But it was sort of forgotten, and mainly because the sequence data was not there to do something useful for it. But then it came back by Martin White and others in like 2008 and sort of. So that was the change of the revolution in structure prediction. So in the beginning, we had contact maps. So this is a contact map here. And this is like what you did before 2008. You had the mutual information or something like that, and you get maybe 20% of the contacts predicted correctly. And you only see that the correct ones are the blue ones, and they are mainly in close to each other in the sequence. They're not very useful for structure prediction. You can use them, they may be used for filtering, but they're not good enough for doing structure prediction. However, if you take into account these indirect couplings, like in PLM-DCA or PsyCOV, you can get like 50-60% of the contacts correctly predicted, and even contacts that are very far away. And then Deborah Marks and others showed that you could use this to predict the structure of single proteins quite accurately. So that's in these cases. However, we should also remember that this is an image. Like in 2008 also there was sort of a revolution in deep learning, and images are perfect for deep learning. They are patterns. And machine learning is perfect for recognizing patterns. So all these machine learning methods that we've done since 2008 and forwards are recognizing patterns. They're basically like trying to look at a part of the map saying, is this a correct prediction or not? Can we enhance it? Can we do things like that? And of course, we see that these contacts has a tendency to be close together along each other. They have a tendency to be along diagonals. So these were our first attempts, and we increased the performance from like 58% to 70% by just a very, very simple, not even a deep learning method. Well, maybe it was a random forest method, but we iterated a few times. So it was very, very simple. And then these methods advanced. We start using more advanced networks. We start using sort of all the methods from deep learning. So this was a UNet in this case. It was our fourth version, PCON-C4. And in these cases, we actually could do predictions that started looking like real proteins. So this was a prediction from CASP-12, I think. And it was like the MSD was 2 angstrom. Today's standard is horrible, but for those days, it was pretty good, actually. And then at CASP-13, we realized that suddenly performance increased. So this is the classical CASP target there. You have all the targets from easy to difficult, basically based on how similar something is to the database at the date of CASP. And then you have GDTTS, which is like basically between 0 and 100, for how similar, how accurate your structure prediction is. So you can see that already, and this is the prediction of the best groups at every CASP from CASP-1 in 1994, so just after I was in China, until CASP-13. So you see, even at the first CASP for the easy targets, which was sort of not super easy, because there was nothing you can do like super simple homology modeling, but it was like distant homologs. Some groups did it correct. And it hasn't changed so much, because you are close to like 90%, which is probably close to how well you can do it. I mean, there was more groups doing it. It was probably, not all groups did it well in the beginning, but then. You see the difficult targets, I mean, there was a jump from CASP 1 to CASP 2, but then basically nothing happened until CASP 13. In the medium one, there was an improvement. So a lot of engineering, a lot of fragment-based approaches, a lot of combination of methods, a lot of ways of doing consensus predictions, a lot of combination of things improved in a quite linear space. Like there was improvement there, but there was clearly something happening in CASP 13. And there was AlphaFold 1, not AlphaFold 2. AlphaFold 1 was basically the same thing as everybody else did, but just better. So there was DeepMind using this DCA and other methods, and then just use a deeper neural network. And then instead of having predicting context, they predicted distances or distance probabilities, and they could turn that into an energy, and they could do a simple simulated annealing or a simple minimization to get the structure. So it was quite good. But so we sat down after CASP 13, thought, okay, now we actually can predict the structure of small proteins. So what is the next step to do it? And it's probably protein-protein interactions. This clearly is the next challenge, because there was a problem for people doing protein docking for the last also decades, and it really didn't work. So we set out, let's try to use the same methods as people use for predicting a single structure to predict the interaction of two structures. So the idea of all these methods is that you have a multiple sequence alignment. If you have two proteins, you need to have two multiple sequence alignments, and you need to align them in such a way so that the proteins that interact are on the same row, which is, of course, very simple if you have one copy of each protein in each genome. So if you put them together, you know that, okay, they are most likely going to interact in the same way in every genome. However, that is not the case for most eukaryotic proteins, because you have many copies, you have paralogs, you have, sometimes it's one protein interacting with many other proteins, sometimes there are specific things, sometimes you go from homomers to heteromeres between bacteria and prokaryotes, etc. Sometimes just interaction changes. So the best guess you normally can do is that you can take like you're just searching of protein A and protein B, and you take the top hits in each genome and hope that they interact in the same way. Certainly it's not always going to be correct, but it's probably the best guess you can do. The problem with that is that you actually throw away a lot of information. And these DCA methods are sensitive to the depth of the MSA. You need to have hundreds or even thousands of sequences in your MSA to do it. And that didn't work always. But at least we could do that. We developed a pipeline to do it. Other people did similar things. So we had a pipeline, we did this combination, we used Tia Rosetta from David Baker, and then we sort of used Pi Rosetta, or modified it slightly to predict the structure of two proteins. And then we did it, and we had some scoring function, which was a consensus scoring at the end. And at least sometimes it worked nicely. So here you have like a predicted contact map on one side and the real contact map on the other one, and they are 80% identical on the interactions. And you have, well, here actually there are two proteins, two copies of each protein. They are perfectly predicted. So in some cases it works. Unfortunately, in most cases it looked like that. Each of the individual proteins were well predicted, but the interaction was wrong. So it was probably as good as anything else at that time, but not much better. We were pretty good at knowing when we were correct. So it had some advantage, but it was not something groundbreaking. In CASP, we had one prediction that was ahead of other people, but in most cases we were not. But then we come to CASP 14. And then we saw this line here. So basically, we jumped from AlphaFold 1, which was the red line, to AlphaFold 2, which was the black line. So suddenly we had the same performance for easy targets and for hard targets. And we are pretty close to experimental accuracy. So and you can see that all the other groups, like the dashed line there, are basically, if you take away DeepMind, so this is basically all the rest of this community that is now as good as DeepMind was 2 years before. So the community caught up, but it took 2 years. So
a few things about AlphaFold 2. So why was it a success? It's not obvious because it's a deep network. It's complicated. I mean, you can think about it. It's not like just throw something in at a standard method. It's really something that someone thought about how to do, and of course tested a lot of things, and some things didn't work. Almost all the things had been proposed before, but not implemented in such a good way. So one thing is that it is actually an end-to-end network. So it starts with an MSA, maybe a template, and predict structure. So like if you have something that can improve the structure prediction, and that affects how you read your MSA, that can be learned. You can also say that most of the information, most of the compute is in the EVA former. So the EVA former has most of the data is there. So and then it's also in ablation studies. You have a structure module that you actually can take out, and you lose some performance, but not very much. It actually you more lose speed than performance. But the EVA former is the key thing here. So what does the EVA former do? So the EVA former takes the MSA. It looks at it using attention transformers, both on the pairs and the rows. So it sort of figures out where you have what pairs should be or columns are important for each other, what sequences in the MSA are important, what sequences can you ignore. It was actually trained with some noise in the MSA, so you added some sequences that shouldn't belong there, just to make the method more robust. That's something that OpenFold realized later. And then it does that, and then you can always go from a single vector representation to a matrix by an outer product mean. And then they do what they call triangle updates. So basically it's like checking if I is interacting with J and then with J with K, so you have this indirect couplings, and you can focus on that attention, so you can focus on the parts that are interacting, so you can learn that. And you keep on doing that 48 times. So basically, you just predict a better contact map, and you iterate 48 times to do it better and better. And it works. It's been shown that this triangle update is quite important. Other parts are quite important. So Rosetta Fold did not use triangle updates in the first version. It used another idea to do instead, and it works almost as well. So then we come to 2021, and if you remember what you did 2021, this is what most people were afraid of then. It was COVID before vaccines. And I was in a very happy time. I was in my summer house, and I had planned to stay on the beach and just relaxing and take it easy. And because it was, I was isolated with my family, and I could work, had a good internet connection. Luckily, I didn't do that because AlphaFold 2 was released. So I did science by Twitter. I was reading tweets. This was the old good time when Twitter was nice. And we worked. But I could work for my doc, and I could have an interview with nature editors sitting in my swimming pants. Luckily, it was not Zoom on, but it was very nice. And I want to highlight here, the key thing for a lot of this is actually the open science. So we really had, this was really Twitter. It was like, I think Min Kung took like 2 weeks or 1 week from AlphaFold 2 code was released until she told the community, the whole world, how to use it for predicting complexes. I mean, it's not that hard. You have to figure out that you have to change one number because you have a residual number, but without Twitter, it would have taken us another 2 months probably. And also, of course, PDB and Uniport was essential because like, without the open data, the sequence structures, nothing of this could have been done. And also was the number of preprints that was coming out this time was fantastic. And also, of course, the license of AlphaFold 2 that was not closed. It was actually commercially available. We could do what we wanted with it. The code was there. It was also amazing. Unfortunately, Twitter is sort of only political anymore, and you can't access it from here. Blue Sky and LinkedIn and other things are not as good for discussions. So we are a bit losing the scientific open communication platforms in the world of division. I don't know. Maybe something that nature can organize, but we should have some good thing there. And of course, the result of this was that not only us, but many, many papers come out within months of the AlphaFold 2 release. It was really for everything. They're using it for DNA, protein, RNA, for using I mean, the first thing that happened was, of course, people solved a lot of structures. People got there with a lot of the data and so on. Speed up a bit. So we went back. We took what we had done for TIA Rosetta and just plugged it into AlphaFold 2. So we had the same way of combining MSAs and putting AlphaFold 2. And I mean, even with default AlphaFold 2, you can get some MSAs and run it together, but you can also pair them in the same way as we had done before. And you can also do like what you call blocked alignment. You just have gaps on one side. And we saw that in our benchmark of protein interactions, we got a significant improvement. So like this is like to the left, is like the Rosetta fold and gram. That doesn't work at all. You basically have nothing correct. This is DocQ, like 0.23 is a good cutoff. AlphaFold 2, without anything, has a few predictions that are perfect. It has an average, 43% of the models are higher than our cutoff. By tweaking the MSAs or pairing them, you get up to 60%. So basically, from something that succeeds in like less than 10%, got up to 60% by just plugging in AlphaFold 2. And we should remember, AlphaFold 2.0 had never seen two proteins interacting. It was completely trained on single chains. So this is not overfitting. This is really trained on seeing new things. Also, that also, of course, also tells you that protein interactions is quite similar to protein interactions within a protein as between proteins. We developed a score for this that could show you that we are good at separating good models and bad models, but also from non-interacting proteins. Like about 60% of the interacting proteins in our test sets could be identified with high reliability. And that enabled us to do some benchmarking on large-scale data sets on input interactions. And here you can see a few interesting things. So this is our predicted DocQ scores, basically how good we think an interaction is. If you look at the red lines there, that's what we get from complexes from PDB. And like 2/3, 60%, we got good scores, 1/3 we get bad scores for. So like 2/3 we can find. Not everything, but 2/3 of them, that's fine. If you take random sets, you see this blue line here, blue dotted line, you basically see that they're all low scores. There are a few high scores, but very, very few. So we are good at separating interacting from non-interacting proteins, assuming that random pairs do not interact. Then you take the black line here. That's from mass spec co-evolution, co-purification studies from Ed Marcotte and colleagues in the UMAP 2.0 database. And you see that it has a clear peak of high-scoring ones. It's not as good as the structural ones. And we can explain that by saying that this is purifying complexes. And of course, not all members of a complex are interacting with each other. Only like some of them are doing that. So if you take complex from PDB, we get a similar scales, like 15-20% of the pairs we got a good score for. However, if you take the green line, we have a much, much lower things. And this is from yeast-2 hybrid data. So in yeast-2 hybrid, of course, we are often seeing transient interactions. So probably interactions that are not part of big complexes, but they're more like regulatory or as temporary. And these we cannot detect. So there's not a strong coevolutionary signal there. So that is one of the challenges that we need to do. We also have recently discovered another challenge. Is actually if you look at homomers, we have a lot of false positives. If you look at the orange curve, that's for heterodimers, and the blue curves of homodimers. So we have find some, but we have quite a lot of high-scoring false positives. And there are several possible reasons for that. One is, of course, that actually homomers are not conserved, so the prediction is not very good. Another one, of course, can be that AlphaFold, and this is AlphaFold 2.3, I mean, it doesn't really matter. They all behave the same way. And that is not very good at protecting homomers. But we clearly have false positives. Yeah, and the positive scores are about similar, so that's okay. But like we have a lot of false positives. And actually, one reason is if you look at this curve. So this is the TM score of homomers, of homologs to homomers. So if you take our homodimers, even if the structure of the individual dimer is almost similar, so basically you have 100% sequence identity, only 60% of the homologs have the same stoichiometry in PDB. Now, PDB is not perfect, and we know there are a lot of modifications and so on, so that might explain that, but it's still quite low. Comparison, if you look for monomers instead, it drops. So if you have 100% sequence similarity, they're always monomers, but then it drops off. So it indicates something that conservation of stoichiometry is probably not very strong. It probably can change quite fast. We have trained a non-deep learning method. It's just logistic regression to try to improve that by using this homology information. And we can go from something like, you can see here, top one, we have quite a lot. On the blue line, there we have a lot of false positives one to much fewer false positive ones if you do this. It's not a fantastic improvement, and it's sort of depending on that you have some kind of homology in the known data set. But we can at least, there are some ways that we can improve it slightly. So that's one thing. Another thing that we have worked on, and also similar to what Trey talked about before, is like we can of course make these networks, and we can find interactions, and you can find different types of interactions. You can look here on the right, you have the proteasome. So you have a lot of, like a big complex, but you also have other type of interactions like the GDI1 RAB protein complex, where there's one protein interacting with many other proteins, but all in the same place. So they can't bind at the same time. However, this proteasome, we can actually build a structured proteasome by doing pairwise interaction and adding one chain at a time. We have to add them in this good order because some are wrong. So Patrick Bryant, my ex-student, developed this small PC program that could do that. And so we can add the whole complex if we do it correctly. And that actually works surprisingly well in some cases, not always. And particularly works well for things with symmetry. So with high symmetry, you can get good complexes. And so this is predictions that are from, this is predictions from a program that has never seen two proteins interacting. And a super simple scoring function, just telling proteins should not overlap. And in some cases, it's pretty good. Skip that. Yeah, and we can also use this as similar to what Trey was saying. We can do it for predicting complexes. It's not as straightforward as you would think. Because there's a lot, in reality, if you look at things, you get quite a lot of noise. So this is trying to predict the egg-sperm fusion complex. So we started with 15 proteins, and we can find that some of them interact with good scores, and some are not. And well, there's one protein that interacts very good. It's Sumo, 1, and Juno, which is known to interact because it's in the PDB. But we can do this, and we can sort of create a network, and we find like a complex of like 8 proteins that might interact together, and then some pairwise interaction outside that we don't know that much about. But if we do that, and we take these 8 proteins and make a model, we can find that only 5 of these actually get good scores if we run them. But it's not consistently good scores. That's something like that. So at the end, we can build a model of a big complex that at least makes some biological sense. It's probably not complete. It's probably not the only complex, but it's at least probably most likely part of the whole egg-sperm fusion complex. And this is a complex that we can, at least nobody has so far managed to purify it because it's very unstable. It's very hard to purify. And it's also hard to express because you can't like have a lot of eggs and sperm growing in your petri dishes. Okay, and then I'll try to speed up to get last year, or AlphaFold 3 was released, and AlphaFold 3 looks like this. Basically looks almost the same as AlphaFold 2. The big difference is that the pairformer, the EVA former is replaced by pairformer, and the structure module is replaced by the fusion module instead, that we can ignore. So the pairformer, the big difference there, they turn around a bit a few things, but the big difference is that they don't use the MSA there. They use a single sequence. So the idea from John Jumper and colleagues was that now we can learn some physics. We can learn to sort of like reason more about the structure by not using the MSA. They have the MSA before. They have a step before they use the MSA. So the MSA is not completely ignored, but most of the compute is not using the MSA. And so we tried this. So first we tried it in CASP 16. And if you look at performance, this is interactions, and you compare to AlphaFold 2. If you take the baseline method, yeah, it's slightly better. So like AlphaFold 3 here is to the right, and you see this purple line, which is like slightly diagonal, so we do slightly better. However, there were people running massive AlphaFold 2 runs, and there you are seeing no significant difference. So like it's, for general protein interactions, it makes a small improvement, but not more than that. However, we went back to the case where it should work better, and also one of the unsolved problems, which is antibody-antigen interactions. And here in CASP 16, well, here it's clear that it works better. So that this is the prediction, the docu score against the number of generated models. We can generate up to 200 models in this case. And if we could find the best model, we see we almost have, in the log scale, we almost have a linear increase of the quality. So if we could detect the best model, and we would have like an average docu of 0.5 if you have 100 models. We also can see that AlphaFold 2 is clearly better than all the other methods. So like even Boltz and Shy, and these are significantly worse. And all these methods increase. However, you can see if we go to the right, if we are taking the top ranked model, not the best model, we are doing much worse, and we are plateauing at something like 0.35 docu. So that indicates that we have a problem of identifying the best models. And why is that? Well, the problem is that the scoring for AlphaFold is really bad. So you have cases like down here, you have the same prediction for AlphaFold, but very, very different models. You have some models that are good, some models are bad. In some cases, not. The overall correlation is not that bad, but for each individual target, the correlation is often pretty bad. And this can be explained by that there are some models that are easy to predict and some that are hard, but even among the hard ones, now and then you get a good model. And then there probably is an interesting set in the middle where there are a lot of both good and bad models. So if we could pick up these best models, we would do much better. We are working on that, and it's some improvement, but not, well, it's significant, but we are far away from the perfect solution. I think we would need to have much more of a structured information to do that. And we also noticed that if you take these models that work well, compared to the targets that work hard, and compare them to the training data set, it's clear that things that are similar to the training data sets, so here to the right, we had a team score of higher than 0.8 or something in the training set, you do much better than for the ones to the left that are hard. So it's clear that this structure model, this performance for AlphaFold 3, has learned something about structure, but most likely it's not physics, it's most likely some sort of memorization of what is in the database. I would say this is not a trivial thing, because if we try to figure out what it has learned, it's not that easy to detect, because there clearly are very subtle things it does. Sometimes it seems like just orientation approach, and sometimes because the interface is completely different and so on, but it's something that has learned. And also you can see that there are cases, even for ones that have no similarity, you do good predictions. So it varies, but it's clearly something that has learned memorization structure. Yeah, and that's it. So I want to thank my people that have done this work. If you want to do a postdoc, I have some openings, PhD students maybe also. And some people pay me for doing this, and they are very nice. So thank you.
Thank you very much. Do we have a question? Yes.
Hi, I'm Tang Tao. My research focuses on the PPI prediction, and I find that current PPI prediction mainly is about the binary classification of PPI. However, beyond the existence of PPI, so different PPI also can carry out different functions, like the activation, binding, post-translational, and modification, and so on. So I think that extend the binary classification to the prediction of PPI type also have great biological significance. However, I find that currently we don't have much data set content that information. I only found the string database. So they contain some PPI types in version 10.5. However, I don't know why they remove this information in their latest version. So do you think that it is worth to explore the multi-prediction of PPI type, and how should we start? Should we build some new data set for that?
I think, so basically you want to ask if for different types of PPIs, if you want to test and benchmark them differently. I think, is that the question? Yeah. So I think that, I mean, clearly protein-protein interactions are very different. I mean, there are different types. and in particular, I would divide things into the ones that are transient and the ones that are not transient. So like transient interactions are, AlphaFold might have some information, but it's certainly very, very noisy. So if you do that. So if you take any regulatory disordered proteins you run to AlphaFold, you might get the right structure that binds, but you cannot use it for distinguishing for true interactions and false interactions. However, for big and part is because of just the physics, because the surface area is small, but it's also because of evolution. Because like to get something to work, you need, for AlphaFold, you need co-evolution. So you need both partners to evolve. And in these cases, for the regulatory things, it's the disordered peptide can probably change very fast, and it can be many different ones binding there, while the target protein do not evolve at the same time scale. And that's the same problems you have for antibody antigens. The antigen is not changing, the antibody is changing. Or for host pathogens, like you have the virus change very fast, but the host does not change. So that's one thing, you don't have the signal there. And that's also because it's smaller and you have less data. So that is, I think, the big difference. And the other thing is, like, of course, you have a problem with AlphaFold is that you have, it works on the MSA. It works on the family of proteins. So you have a lot of false positives to say that these two families interact, but it doesn't mean that these two individual proteins interact, because it can be, and you get a signal for the whole family. So if there are point mutations that make two proteins not interact, the current versions of AlphaFold do not separate these. And to solve that problem, you probably need something that actually handles the atomistic representation of a protein much better. So I think we can get there, but you need to get into the physics somehow, saying that you have understanding that better. Did that answer your question?
Yeah, I'm not pretty sure because you mentioned the transitant type. So that's actually that's another question that I always think. So as you just said, you just mentioned the transitant type, and so why I just talked about the activation binding. That's kind of the protein PPI actions, the actions of protein, or kind of mode of protein. So that is the PPI can be classified, can be divided into multiple types based on different principles. So I think that maybe, so sometimes I think that can form a more complex problem. But I haven't.
Yeah, certainly in the cell it's more complex, but at the end it's back to atoms that interact. So like at the end it's always something, but of course you always have long different time scales. You have, of course, not only two proteins. That's simplifications. We have many proteins that interact together, and of course we have a lot of other non-protein molecules. Also, you have ions, PTMs, nucleotides, and so on. So I think so. You're absolutely right saying to say it's more complex. And it's, I think, at the moment we rely on the evolutionary signal. That's what we have, and all these methods that are trying to do it on the physics or atom-based are not doing that well, to be honest. They do well on things that are in the database that are trained on, but for novel things they are not showing great progress yet. They might do it in the future, but not yet.
Yes, thanks a lot.
Thank you.
I think we need to move on. Thank you, Dr. Ellefson. Our next speaker is Mingkyung Baek. She's coming to us from Seoul National University. She has very strong interest in the application of AI.
In this era,
structure,
what are the missions of a university?
actions of proteins.
What actions should we take?